Today we're going to be doing a lab that's going to be measuring the rate of photosynthesis. So we've already split you up into different groups. Some of you are going to be doing bright light with a baking soda solution. Some of you are going to be doing just the room light with the baking soda solution. Some are going to be doing no light. So your apparatus is going to be hidden underneath a box and you'll check it every 2 minutes. Again with the baking soda solution. And then others are going to do the same sort of setup, only you're just going to be using water without an additional carbon source. Okay, so at each station you'll need to have a small cup like this, and then you're going to pour out whichever solution you're supposed to be using. Now I'm going to show you with the bicarbonate solution, the baking soda solution. So I'm just going to pour a little bit of this into my cup. That should be plenty. And then you should also have a 10 milliliter syringe, looks like this, and a hole punch, which looks like this. Okay, so to begin, you're going to just simply remove the plunger from your syringe, and then you're going to take a spinach leaf that looks like this and your hole punch, and you're just going to punch out 10 little holes, but you want to avoid the big veins that are in the spinach leaf. So you're just going to run this through here. Okay, and punch out 10 little leaf punches. So you'll notice I try to go along the edge. So that's where you're going to avoid those large veins. Okay, and you just need 10 of these. So let's see, I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay. So you're going to place those leaf discs into the syringe. So you're going to take this piece here, and you're just going to drop your leaf sections, your little hole punches of spinach leaves, and drop them down into your syringe. And then just give them a little tap to get them all down to the bottom. Okay, so just sort of tap it and they'll all come down here to the bottom. Okay. All right. Then we're going to replace the plunger. So we just place that in. Be careful not to squish your poor little discs, okay? So you might need to leave a little bit of room here to begin with. All right, then you're going to place the tip of the syringe into your designated liquid. Again, I'm using the bicarbonate solution, and we're going to draw up 5 milliliters of solution. And I like to go a little bit past the 5 milliliter mark, because if you'll notice, I've got a lot of air trapped in here, okay? So I need to get that air out. So I'm just going to tip it up, and I'm going to try to get that air bubble out. There we go. Okay, and then I just need 5 milliliters. So I'm just going to get rid of the excess. Now all I have in here are my little leaf dots and my 5 milliliters of solution. Okay, now you also have this little stand. Okay, and you can use this if you want. I'm just going to use my finger because it really doesn't hurt that much. You're just going to be creating a vacuum inside this plunger. So what we need to do is we're going to be getting rid of all of the air inside our leaf cells. So I'm going to cap the end of my syringe with my finger, and then I'm just going to pull back on the syringe a little bit, and you'll notice all of those air bubbles that come out. So I'm just going to sort of tap that, then I'm going to gently release my plunger, and I'm going to try to get rid of those air bubbles. Okay, so I'm just going to give a little depression there, right? And you may have to do that a couple times. So we're just trying to flood the leaf tissue with this solution so that we get all the air out. So again, I'm going to cap it with my finger. I'm going to pull back, and you see those air bubbles. Okay, so we're going to tap again. Okay, Okay, tap, 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 tap. Try to release those. Gently release your plunger. And the air bubbles will go to the top. And again, we just sort of push that out. So eventually, we're gonna see that our leaf discs are gonna start to sink, okay? And that's what we want. So you may have to do this sort of plunge and tap a couple times, okay? There they go, they're starting to sink. So I'm gonna do it just one more time. Okay, pull back. Tap, 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 tap. Okay, gently release. And then get that air out of there. Okay, so let's just do a quick check. We really need all of those leaf discs to sink, and I think they're all gonna go this time. Yep, there they go. Okay, so that's where you want as a starting point. You want all of those leaf discs down at the bottom. So again, you may need to do your infusion where you just sort of plug the hole, pull back on that syringe, give it a tap, and then release that air so that you have all of those discs nice and sunken. Okay. Then what we need to do is we're going to pull enough liquid so that we've got 10 milliliters of our solution. Okay, so I went right back to that same solution. 10 milliliters goes in, and go ahead and just leave your syringe up. We need those discs to sink. Okay, so I'm just gonna leave it. There we go. So we're gonna set this up on our desk, and those of you who are just in the natural room light, you're just gonna leave your syringe just like this. Those of you that are in no light, you have a big box that you're gonna bring and set over the top of your syringe. And then you're just gonna take the box off every 2 minutes so that you can check your discs. If you're in bright light, there's a lamp that's set up at the side of the classroom. You need to place your syringe directly underneath that bright lamp. Okay, so make sure you're setting your syringe in the right place and that you know which syringe is yours. So maybe leave a little post-it note that's on the counter, right? Leave a little post-it with your group's name. And all you're going to do at this point is just time it. So you're going to be timing it every 2 minutes, and then you're going to come over and you're just going to give your syringe a little tap like that, okay? And you're looking to see how many of your leaf discs are floating after each 2 minutes of timing that you're doing.